Okay, so thank you for joining us tonight at the Prince William Sound Science Center's weekly lecture series. Before we get started, I just want to point out some information on the back table. There is information there for a class. There's actually two classes of information. One is for a fisheries technology class that's being taught here remotely by a local fish and game biologist, Tommy Sheridan. Some of you may know him. So he's teaching a class that's going to happen this coming semester, and it will start in January. Registration is open now with Prince William Sound Community College. It's actually being taught out of University of Alaska Southeast, but it's being sort of networked here in Prince William Sound through the community college. So it's 4 credit, and it's part of a larger fisheries technology program. And members of communities, rural communities, as well as native members, can enroll for free into this program. And you get a degree at the end of it. It's a number of classes. So there's information about this fisheries technology program, as well as the class that Tommy is teaching this coming January. And there's also information back there about a class for high school students for fisheries technology as well. So if you know of anyone who's in high school that has an interest in resource management, fisheries, like going to work at one of the aquacultures, working in one of the canneries, this might be a really good class for them to take that they can get extra credit. So there's information back there. And with that, I'm going to turn this over to Megan Milligan.
I'd like to introduce our speaker tonight, Charlayna Cammarata. She works with us at the Science Center as a research technician. She came to Cordova in August from NOAA's Kachemak Bay Research Lab, where she did some work on some of the work she's going to talk about tonight. She graduated from Alaska Pacific University in 2012 with a BS in Marine Biology, and she plans to attend Texas A&M University at Galveston. So, please welcome Charlayna.
Hi. Just a little bit more brief background on me. I'm originally from St. Louis, Missouri, and I moved up here in 2008 to get a Bachelor of Science in Marine Biology from Alaska Pacific University. And are any of you familiar with that university?
Yeah, down the coast area.
No, it's in Anchorage.
Oh.
It's okay. Well, anyways, it's a small private liberal arts university in Alaska. It's the only one. And fortunately, it allows students to be able to get out and get some real-world experience at the same time as you're getting your degree. So I was able to land a NOAA internship that was also partnered with USGS, and I worked on sea turtle disease in Hawaii for a summer, and it was awesome. And I was accepted for a second NOAA internship, and that was this one in Kachemak Bay, and I worked with Chris Holdreed out of the Kachemak Bay lab down in Kachemak Bay. And I worked on how oceanography and paralytic shellfish poisoning are interrelated. So I'm going to discuss a little bit of a background on phytoplankton, just kind of see where everyone's background is on, and then talk about the issues with paralytic shellfish poisoning, and go into some of the methods that we worked on and some of our findings and what it means for here and for all of Alaska. So I can take questions anytime. I have a question slide at the end, but if I know with me, like if I don't ask my question right away, I forget it. So.
What is this shellfish poison? A virus, bacteria?
I will get into that, but it's caused by a phytoplankton. It's caused by an algae cell.
Algae. Now, how big
A plant cell.
How big are those relative to, say, bacteria?
They're larger than bacteria.
Larger than bacteria?
Yes, yes. But I will get into that. So here are the collaborators that worked on this project, myself and another intern, and these 4 people all work for NOAA. So, any ideas on what phytoplankton are? They are plants. Since you brought up zooplankton, what are zooplankton? They're animals. Okay. Anyone else have any ideas on what phytoplankton are? Uh-huh.
Does that mean it has photosynthesis?
Yes. So phyto means plant and plankton means drifter. So they're microscopic algae cells, microscopic plant cells that float around in the surface of the ocean or lakes or any kind of body of water. And these are 5 pretty common types. And tonight I'll be focusing on dinoflagellates, which can be harmful. But a lot of these are perfectly normal. Phytoplankton actually make up the same amount of oxygen in the atmosphere as our tropical rainforest. So everyone's like, save the rainforest because of the atmosphere, but you also want to save the phytoplankton too. And not a whole lot of people notice that. Anyways, they're primary producers. They photosynthesize, so they're the beginning of the food web. There essentially are grass and trees and things on land, but they're in the water. And they feed zooplankton, which are microscopic or macroscopic fish larvae or crustacean larvae or whatever that also can't swim very well. Dinoflagellates and also diatoms, those are the 2 most common here in Alaska. Some of them can be harmful, and when they're in bloom, we call them harmful algal blooms, or HABs. And so I was working with harmful algal blooms, specifically with Alexandrium, and I'll get to him in a minute, but Alexandrium causes paralytic shellfish poisoning. So this is what an Alexandrium cell looks like. And this is the flagella. Dinoflagellates have flagella, or a little tail. And it comes down here, and it can kind of move itself, but not really. The waves and the tides kind of push it around a lot. And these are very, very small. You can't see them with the naked eye. You need a microscope. Even with a microscope, they're hard to see. And so why should we study Alexandrium? There are a lot of human health concerns with Alexandrium. Most species, there's I think 20 some odd species of Alexandrium. I believe 15 or so of those produce saxitoxins, which are neurotoxins. They shut down your nervous system when you ingest them. Well, not always, but if you react to them, then they will. And this can cause, if someone were to ingest shellfish that have paralytic shellfish poisoning, they could end up in a hospital and all this other stuff, incur costs, etc. Things that you don't really want to do. You really just want to enjoy your mussel soup. Lost revenues. Little known fact, I guess, for most people, which is interesting to me. Alaska spends at least $10 million a year to prevent people getting sick. Just on testing shellfish, they spend $10 million a year. That does not include when beach closures have to happen. So if an area tests positive, they will close down the entire beach. Even if the entire beach isn't infected, they'll close down the entire beach and no one can go out and collect. And if you do collect, you might get sick and the state is not responsible.
So they are responsible then?
Well, if they tell you not to collect and you go out and do it anyway, then they're not responsible.
So if we go out and get sick, we have to pay the bill, right?
No. I don't know. So, have any of you ever been to Kachemak Bay? Yeah? Yeah? I love Homer. It's one of my favorite places in Alaska. So, down here is South Central, and this is Cook Inlet. And this little box is blown up here. So, Homer's right here. This is the famous Homer Spit. And Seldovia is down here. And the Kasutsna Bay lab is actually right in here inside of Kasutsna Bay. And we were studying all throughout the bay. So why do we study Kachemak Bay for paralytic shellfish poisoning? Because there's a lot of shellfish. Kachemak Bay holds, I believe, 14 commercial oyster farms, 1 commercial blue mussel farm, and I believe soon to be a geoduck farm. At least there was some talk about it. commercial as well. So they get tested by the state. If it's a recreational beach, which there are a lot of those too, they don't get tested regularly unless there's funding for it. But there's lots of butter clams and little neck clams and mussels that people go clamming for in Kachemak Bay. And these guys are clamming for razor clams in Inilchuk, but that was the closest picture I had. So the goals of this project were to figure out if Alexandrium was present in the bay. We had no idea. Kachemak Bay has a pretty low prevalence of paralytic shellfish poisoning in general. So is Alexandrium even there? Should we be worried about it at all? Why should we be monitoring for HABs there if there aren't any? So if it is there, where and when? Where in the bay and when in time and in season? And under what oceanographic conditions do these occur? I also looked at different sampling protocols. I'm not really going to get into that, but if you're interested, go ahead and ask a question. So we had a few hypotheses, and these are based on previous studies in different places, because there's never been a study like this in Kachemak Bay. So here we have a column of water. So this would be the surface of the water, and this would be to a particular depth. And in this column of water, this is perfect environment for Alexandrium. We have fresh water flowing opposite of salt water. It's called two-layer flow, and it allows for a stable environment. The stable environment creates a perfect environment for the Alexandrium to circulate between nutrient-rich water and salinity or less saline water near the surface to be able to photosynthesize and stay in a similar place so they can bloom. So we expected to find Alexandrium in these places that have these stable environments. And we classified stable environment as relatively warm. 10 degrees C is not very warm, but it's kind of warm in a place that has lots of glaciers. Mesohaline salinities, so we want intermediate salinities. 24 to 28 is intermediate. Normal seawater is about 35. Ambient sunlight, lots of nutrients, and other stability promoting factors, which could vary depending on where you are in the bay. It could be ocean circulation, little gyres, or if there's a submerged like rock face or something, that could cause some issues. So basically what I did was I sampled from the end of May until the beginning of July. Much more sampling has happened since then. I just wasn't there for that long. And I sampled in two ways. I took a 10 liter sample from the surface. Usually it was 10 liters. It could be different. And I took that with a bucket, and then I filtered it through a net. And then I also dropped a net to a particular depth and pulled it up. And I recorded all these different variables to be able to start answering these questions. Then I came back to the lab, and I looked at these samples in two different ways. I split them in half, and I looked at them under a microscope, and this is actually a picture of what our microscope looks like with all the samples in there. These are all diatoms. There's probably, that might be a dinoflagellate right there. So I would go through and count every single cell, every single one of these little guys in the slide 3 times to figure out what was actually in the sample. And then I would extract DNA and try to find the concentration of Alexandrium. And I went to North Carolina to run all the genetics, because we have a sister lab in North Carolina, in case you didn't know. So these are my results. So the yellow dots are where we actually took our samples from, and they range all across the bay. And these black triangles represent the relative locations of the oyster farms currently in the bay. And there's also some recreational shellfishing in here, also over in here, China Poot, and Bear Cove. So these pink dots, yes?
The recreational, was that where you go butter clam digging then?
Yeah, you can go, I know people go butter clamming here in Kachemak, or in Kasitsna, and back here in Chinapoot. I think I think it's Little Necks. No, I know it's Little Necks up here. Probably Mussels and Little Necks in Jackaloff. But I know there's a big butter like pit in Kasitsna, and there's also a big one back here in Chinapoot. So all these pink dots are where we found Alexandrium. Just every site. Of the 70 samples that I took, only 5 were negative. But we found them in extremely low concentrations. We only found 4 samples, these 4, that were actually really high. But they weren't high for very long, so the shellfish never got sick. So this graph is just showing the relative concentrations that I found with genetics versus the date. These 4 samples, the highest concentrations, above 100,000 cells per liter. That's a lot. That's every single little microscope slide. I should have seen 100 or more cells of Alexandrium to be able to see these. Unfortunately, I didn't see any Alexandrium, and that's because they're really hard to find. When they're in really low concentrations, there's just not enough of them in a sample. You'd have to look through the entire sample, and that's kind of impossible to do when you collect 50 samples in a day. And these 4, I should have been able to see under a microscope, most certainly. These 4 were cause for concern. But I am not a trained phytoplankton expert. I was trained for about 3 days on how to do all of this, and when I saw stuff that I wasn't sure, I was like, oh, I think this is Alexandrium. I would call some experts, and they were like, well, I don't really think so, but since we're only seeing pictures and they're not alive and all this other stuff, it's really hard for us to tell, so we think it's this one, or we think it's this one. It's not that it's a bad method, it's that it's a difficult method, and genetics seems to work a lot better. And I like genetics. It's cool to wear a lab coat. So here we looked at the effect of salinity on the abundance of the Alexandrium, and we found Alexandrium fundiensiase in the bay, or that's what I screened for. And there's another species that's pretty common in Alaska, it's Alexandrium ostenfeldii. I didn't find any in my samples. But anyhow, random aside.
Charlayna, how difficult is it to distinguish between the species?
It's extremely difficult. You have to, this is going to take me forever if I go all the way back. Do you remember the picture of the Alexandrium? And it had those little, like, scale-looking plates? So the arrangement of those plates tell you what species it is. And you have to have a microscope that can see that much. Our microscope was just a really crappy old, it was probably a 50-year-old Nikon that was half broken. I could only go to 40x or 400 times magnification. You really need something that goes a lot higher. And you need something that has a really clear view, both underneath and above, which ours didn't. Our lenses were really scratched up and everything, and just kind of that's what we were dealt with. But they're very difficult to distinguish, and a lot of times people have to look at them under different types of microscopes, like scanning electron microscopes and other things, to see them in different colors, to be able to distinguish what species they are. So the effect of salinity, as we predicted, the highest concentrations fell in between this 24 to 28 range, which was really exciting. And then for temperature, the same thing. The highest concentrations were above 10 degrees Celsius, which was also really exciting because no one's ever looked at Alexandrium in this bay before. And so we didn't know if they would even act like they do in California or in England or Scotland or Maine. And so now we know, this is sort of validated that all the literature that I was able to look at also agrees with here, so we can use the literature to start cross-referencing what we're finding here. Something interesting that I found, one of the more interesting results, was the ebb versus the flood tide. So the flood tide is when the tide is coming in, the ebb tide is when the tide is going out. And on average, we found that the ebb tides contained more Alexandrium cells than the flood tides. This really boggled us. We were like, shouldn't there be more coming into the bay when the bay is flushing in and all this water is coming in from Cook Inlet and the Gulf of Alaska? Like, it would make so much more sense if it was coming from out here and going in during flood. But this is indicating that instead Kachemak Bay is a net source of Alexandrium and it's getting flushed out of the bay during the tides. So to start looking at whether or not this trend is actually something real, we've tripled up our sampling and we're continuing to sample every couple of weeks, every couple of months. Yes?
Is there a way that it's sort of being concentrated as it's flowing out? If it's flowing in, then you've got all this extra water behind it and diluting the phytoplankton maybe?
Possibly. So I did mention glaciers earlier. There's actually a huge watershed up in this range. I know I'm on the ceiling, but up in here is, what is that river? Oh no, I can't think of it. It's a giant salmon river. Salmon. Maybe I'll remember it in a few minutes. So there's this huge watershed that floods into the bay. There's also a huge glacier field that has, I think, 13 glaciers in it up here in the Kenai Mountains. And I would assume that as the water is getting flushed out, it would also be getting diluted because you're getting all that extra water from land sources. But Alexandrium is really good at concentrating, and it has to do with their life cycle, which I can go into. I have a slide on it a little later. But Alexandrium are able to form cysts. So if the environment isn't favorable for them, or it's starting to get cold and they want to shut down for winter, they basically roll up into a little ball and sink to the ocean floor, and they just sit there until conditions become favorable again, and then they hatch. It's like chicken.
Is it the Russian River? Is that the river that flows out?
No. Fox River. It's the Fox River. Fox River Flats. Okay. Sorry if this is a little difficult to see, but I just wanted to show you the differences between location and the same temperature and salinity graphs. So we have green for Kachemak. So Kachemak samples are right here. They're just out in the middle of the bay. Black is for areas that have glacier input, and we have all of these samples plus one down here. And then non-glaciated are these brown samples, and I think they're the hardest to see, and they're down here. It's not an even split. We kind of did what we could. We're working, actually our sampling plan now has much more in Kachemak Bay and more of these non-glaciated areas to start looking at these issues too. So temperature, the glaciated areas actually have the highest concentrations, but the trend is still, it seems like with temperature at least, it doesn't really matter where it is as long as it's the right temperature based on how these, how distributed these colors are. How are glaciated areas warmer? This is my project, Becca's project, Rebecca Holman. She was the other intern. She worked on all the CTD casts and things that go along with this. She found a lot of really interesting data with the CTD cast in that the salinity would let's see, it would be super fresh and then go super salty and then go back towards more fresh the further down you got. And we saw that profile like the whole time we were sampling all summer. And with the temperature, it was very gradual. We have very stable environments in Kachemak Bay. Just some areas, if they're really well mixed, they'll just be a straight line up and down, vertical line. We had a couple of those, but not very many. Most of them were very warm at the surface from the sun and other things, and anywhere from 5 to 15 meters down, shot down to really, really cold. So I don't know if that's because of the glaciers, but that is the trend we did see. This is the temperature at the depth. So whatever these are, it's the temperature at whatever particular depth the sample was taken. So these four are actually surface samples. We got the highest concentrations of surface samples. Some of these over here were also surface samples, but some of these they could have been 10 meters deep, depending on where the sample was taken from, the phytoplankton sample. For salinity, we see a little bit more of an interesting trend. Again, the glaciated ones have the highest concentrations, but there's less of a spread. It looks like the non-glaciated areas they're just too salty. They don't get enough freshwater input to be able to get to these concentrations for us to be of concern. That's not to say that it won't happen. There was a woman who lives in Kachemak Bay, and in March of this past year, she got very sick because she ate a tainted butter clam harvested from Kachemak Bay. Problem with butter clams is they can hold on to toxins for 2 years. So you're not sure when that butter clam got infected or ingested that Alexandrium. But it did cause some problems, and a lot of people were very concerned because that's never happened before on this beach. So it can happen in those non-glaciated areas, but at least the data that we have, it shows, or it's telling us that maybe it's not very favorable of an environment, whereas the glaciated areas are probably more favorable. So I had a lot of people to thank. The Office of Education helped put together this internship so I could do it, and I got paid. It was pretty nice. And then I worked at the Kachemak Bay lab, which is a beautiful lab if you ever get a chance to go. And then I worked at the Beaufort lab in North Carolina. This is the sister lab to the Kachemak Bay lab. And this is the Research Reserve / Islands and Oceans Visitor Center. And they run the harmful algal bloom monitoring program in Kachemak Bay. And I had some help from a few other people, another NOAA office. Ray Roland was really helpful with all the shellfish information. And yeah, that was about it.